All right, welcome everybody. It's a pleasure to see you all in the attendance box.
Welcome everybody.
I should
It's a
probably
pleasure to see you all
feed off so that you can't hear me twice. We're live and direct with Upinder Bhalla today as part of the Neurotheory Forum online and also as part of Worldwide Neurotheory. Follow us on Twitter and follow us via the website. It's my distinct pleasure to have Upinder Bhalla, Professor Upinder Bhalla today here to tell us about his science and the science of his lab. I should quickly introduce Upi. He is probably for me one of the most prominent figures in neuroscience anywhere other than the United States and Europe, in that it's really difficult to, for me, to meet anyone, especially from the sub-Indian continent, from the sub-Asian, from the Indian continent, from the Indian subcontinent, I suppose, who hasn't somehow been propelled forward or been in touch, or whose science has been promoted by Upi. So he's visible not only through his science, but also through all the people that he's been supporting in their quest to stay in neuroscience, I suppose. And then it's also really impressive to see the many different creative approaches that are coming out of his lab in using tools that are not traditionally being used for experiments. For example, quite recently doing imaging and uncaging experiments with projectors, for example, and non-traditional equipment. So there is a very neat sort of non-conventional approach to many of the things he does, and I like that a lot. Upi has been an undergrad at the Indian Institute of Technology in Kanpur, as well as the University of Cambridge in the UK. He then got a PhD in biology at the Institute of Technology in California, Caltech, and did a postdoc at Mount Sinai in New York City. And after that, returned to India and became a reader at the National Center for Biological Sciences, where he's been ever since. And he's made up his way up the ladder, becoming a full professor in 2008. As I've already said, Upi has raked in a number of awards that are quite extensive in their list, so I won't tell you. And instead, I'll let Upi introduce his science that can be, or that is introduced today as, I guess, the revenge of the butterfly. So thank you very much for being here with us today, Upi. I'm looking forward to your talk.
Thank you very much, Tim. And it's a great pleasure to be here at the Neurotheory Forum. I hope that everything comes through clearly and with appropriate visuals. We're having a lovely thunderstorm here, and I am connected through a mobile phone. So perhaps the occasional flashes of lightning will interfere with the transmission, but I hope it won't happen for too long. So I'm going to, oops, let me get back to the beginning of the talk. I'm going to talk about something that's very dear to my heart. It's something I've been thinking about pretty much ever since I got into neuroscience. And that is the kinds of computations that neurons can do, that single neurons can do. And I really love this quote from Ramon y Cajal, because in it he has glimpsed at what I think to be the essence of what neurons are in terms of their role in the nervous system. They're not little black boxes, they're not little points. They're beautiful, complex structures with very, very elegant architectures and amazing, I think, amazing computational functions, which I will try to convince you about. And so, yeah, to quote him, the mysterious butterflies of the soul, the beating of whose wings may someday, who knows, clarify the secret of mental life. So with those words, let's launch forward. So I'm going to say the butterfly strikes back, neurons doing network computation. And I think that's pretty clear that I think that some of the things that people ascribe to networks are actually being done at the level of single neurons. Okay, so here's an interesting comment about the way people have thought about the brain and the mind, which is basically that the most advanced technology of the day has been the primary metaphor. So let me just run through a few of these. Here, there's, of course, mechanical metaphors, like this orrery, this mechanical solar system. But here's one of the oldest metaphors for the brain, and we still have echoes of it in the terms that we use to describe people's disposition. So phlegmatic or bilious or sanguine, these are all bodily fluids, actually. And they are derived from the old hydraulic metaphors for how the brain and mind worked. This is an absolutely gorgeous tapestry, and it illustrates one of the other metaphors. The shuttles weave a dissolving pattern, always a meaningful pattern, though never an abiding one. This is by Charles Sherrington. So it's one of the examples of the metaphors involving mechanics. But I like this particular tapestry because if you look closely at it, it's got some what look like little circuit diagrams and networks on it. So this one thing has multiple metaphors, so to speak, in one image. Here's, of course, the metaphor that many of us are totally familiar with, that of the brain as a computer. And there's some lively debates about how accurate and otherwise this may be. I won't go into those today, just to say that this is one of the common metaphors that we use. But here's a metaphor that's been around actually for a very long time. And back in the good old days, when I was but a lad, this was in fact one of the metaphors that I ran into when reading about the human brain, that of the network, the telephone or the telegraph network. And interestingly, that has now become one of the key metaphors for many things today, because networks are all pervasive. There's the internet, there's social networks, there's gene networks, and so on. And somehow this seems to have been taken up as one of the major ways of thinking about the brain. For example, the essence of the connectome projects is the idea that by working out the network structure, the circuit diagram, you will know most of what needs to be known about the brain. In fact, there's a very nice TED talk by Sebastian Sung saying, "I am my connectome." And although I don't necessarily agree with all the conclusions, it's a very nice talk, and you should have a look at it if you haven't already. So all of these have to do with networks. And what I'm going to do next for a few minutes is to talk a bit about some of the kinds of computations that happen in networks. So here is a typical feed-forward artificial neural network. It's got an input layer, it's got a hidden layer, and an output layer. And each of these round things is what you could loosely call a neuron. But the soul of the network, if you like, is in all of these connections, their projections, their weights, and so on. So what can this do? This can do many things. For example, many years ago, Terry Sachnowski trained one of these networks in the old days of neural networks when they were just small, they weren't deep yet. He trained it, made an application called NetTalk. And what it could do was to take letters of English text. And of course, English has absolutely no logic when it comes to how you actually pronounce things that are spelt in a certain way. But what this neural network did after sufficient training was that it managed to figure out how to convert the letters, which are in the whatever random form that English spelling adopts. It managed to convert it into correct phonemes most of the time. And the kinds of mistakes it made were also instructive. It made the same kinds of mistakes someone reading a word but never having heard it might make. In fact, kinds of mistakes I have frequently made. So it would make the same kinds of learning and same kinds of mistakes and same kinds of ability to generalize that that real neural networks do. And this is, even in those early days, I thought this was extremely remarkable. I thought this was telling us something very important about how the brain might function. Here's another kind of network. It's different from the previous one. Note that this is a feed-forward network. It only goes from one layer to the next downhill, so to speak. But this one has a bunch of inputs, and each of these are neurons. But the neurons, as you can see, connect back onto themselves. So this is a recurrent network, and this one forms an associative memory. And it can do various things too. It can handle degraded input. So all of you looking at this image should be able to recognize blurry and out of focus waves, which is what your neural network can do. And that's what this kind of network is good at. It's also very good at doing pattern recognition and completion. So for example, if you train it up on a couple of patterns, let's say this funny face here or this random pattern here, and if you then provide it with only partial input, after a couple of cycles, it will regenerate, it will complete the original pattern. So this input is enough to tell the network that really that is the pattern that it learned. And remarkably, this kind of scrambled thing is closest to this one, believe it or not. And so it, again, converges to that. So these are the classic forms of what neural networks can do. But about a decade or so ago, what happened was that totally absurd amounts of data started to pile up in all the big internet companies, Google, Amazon, and so on. And totally absurd amounts of computing power became available to them. And so what they did was something which the early neural networks people could not do, which is they started to be able to train networks with huge numbers of layers, hence the word deep networks. They're deep in that they have many, 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 many layers deep. And surprisingly, at least to me, it's surprising how much more you can do once you have the ability to go deep into the neural network. And I'll just give you an example or two of this. So here is one of the classic examples from NVIDIA group, Karas et al. And what they have in this is a bunch of faces. These are the reference faces. These are trained faces. And what they have here is a bunch of faces, and the neural network is asked, take the trained faces and map them to this destination face. And I cannot tell that any of these is a non-human, but all of the ones in this grid of 5 by 3 are actually entirely computer generated. I find it astonishing, especially, for example, they've ornamented this face with a beard, though of course the woman on this side does not have a beard, and nor does the man here have a beard. Anyway, you can do quite amazing things with these networks. So now, one characteristic of these networks is that they use very, very simple neurons. These are basically simple weighted summating devices. They take a bunch of inputs. Each input has a weight associated with it. And you just sum all of these together. And it goes through perhaps a nonlinearity to give you your output. Right? So this is, to my mind, an embarrassingly stupidified version of a neuron. And yet, these networks can do these things. So this then, of course, brings me to my core interest, which is, okay, this is what you can do with really, really stupid neurons in networks. Let's think about what happens with more realistic, computationally capable neurons. So let's come to our one of our first heroes of this, which is Wilfred Roll, who sadly passed away a couple of years ago. So the soul from Bhalla aren't simple spheres, they're not point objects. And through a long career, where at first actually he was quite ignored or even scorned, in that people didn't believe that it was important to have all these branches, he methodically went about working out the biophysics, calculating the effects of the branches, and thinking about the computations that might emerge from there. And he did some really remarkable calculations as well as mathematical analyses. And I think he has played an enormously important role because, of course, he was right, and now people do appreciate what neurons can do, even though they aren't used to their full extent in neural networks. Okay, so let's take Roll's contribution as a given, and just think a little bit about what that means when we look at the brain as a whole. So this is an old hierarchy diagram saying that when you want to understand something like this, when you want to understand the brain, you really need to think about its functioning at all of these levels. And the problem I feel with many neural network kind of analyses is that they sort of hover around here and don't give, let's say, due respect to the kinds of stuff that is happening over here, which I will argue in a little bit is perhaps most of what is happening in brain computation. Just to terrify you, here are a few equations. This is what I unleash on all the students in the courses I teach. And these are basically the equations more or less pertaining to the levels of description that we have here. There's various chemical equations, diffusion equation, Nernst equation, cable equation, Hodgkin-Huxley equations, and an equation for synapses. I find it remarkable just how much can be explained with this small set of equations. And most of what I'll be telling you about today is simply within this very small set of equations. So it's not that neurons are complicated in the physics or in the chemistry, but they are very complicated in what they do when they put all of these things together. Okay, so just checking that things are connected. All right, so just some other background information, some of the typical numbers in the brain. There's roughly 10 to the 11 cells in there. There's roughly 10 to the 15 synapses. Each synapse has a huge number of reactions taking place, so you can think about what kind of complexity that means. Electrical events are fast, chemical events are relatively slow. Structural events, that is changing of connections, can be slower still. There's an intriguing problem, which I won't get into. Lifetimes of proteins are relatively short, yet the neurons that are made of the proteins last your lifetime, and so do your memories. So how do you manage to do that? Well, that's a story for another day. And here's just a listing of scales from what's happening at the molecular level to the dendritic level, to the cellular level, and then up to the neuron. Okay, so let me start by stirring the pot a bit. I will give a provocative, I hope, view on what different levels of brain computation can actually accomplish. So what network computation is very good at is it summates input over a very wide range spatially and a wide range of modalities. That is, it can take inputs from all your senses as well as internal representations and generate, of course, many kinds of motor output. Its functions of, well, if you believe the hardcore network point of view, its functions are everything. It controls all the major systems levels, activities, sensory, internal states, and motor control. And one could argue that these are all high-level computations. At the electrical level, we have rapid summation. The length scale of electrical propagation in a cell is of the order of 0.5 millimeter. It's very good for thresholding and amplification, and it's very good at fast, long-range transmission. which is why we can be of the length scale of meters and still have relatively good control over the mechanics of our limbs. There is very fast neurotransmission. So these perhaps do somewhat more elementary computation. At the chemical level, you have actually very fast computation when you talk about synaptic transmission, which is mostly chemical. That is the millisecond range. But some of the slower things are spine computation, where we'll see it transforms a lot of complicated inputs. It does these things in extraordinarily small volumes, including the dendritic spine. You can make genetic decisions chemically, and long-term storage is handled. Memory, in other words, is handled at this level. Housekeeping is something which is seen as not very interesting, but actually it's remarkably interesting and complicated. And these are also regarded as elementary computations. So fine, so this is not yet provocative. Let me get provocative now. I'm going to put some numbers to these. I'm going to say that, oh, well, network computation is of the order of 100 milliseconds. So that's if you have good reflexes, you can typically respond to things in the order of 100 milliseconds or in that range. The dense size of networks is reasonably large, depending on the scale of networks you're thinking of. And the number of areas in the brain is in the order of hundreds. So if you were to put all of these numbers together, you would say that there's, maybe a 10th of a million operations happening per second. Okay, that's not too bad, but let's see what you can do when we go down to the individual neurons, to the electrical calculations. So these are fast. They're of the order of a millisecond. Because the length constant for electrical propagation is quite long, so you have roughly 10, in a large cell, you have roughly 10 different electrical zones, because the length constant is about 0.5 millimeter and the dendritic extent is about 5 millimeters. There's a lot of cells, and you put all of these things together, and you end up with something of the order of 10 to the 15 operations per second, which is a big number, but in the modern world, it's amazing how things catch up with us. So these are now numbers which you can actually imagine doing on a supercomputer. Okay, chemical computations. Let's concede that maybe we should not count synaptic events. So these are of the order of, so let's just say that chemical operations are of the order of 1 second. And checking that things are going. Okay, so chemical computations of the order of 1 second. Because the diffusion constant limits how far signals can propagate, the cell is much more finely subdivided. So basically, every single dendritic spine is like a chemical compartment. It has a local set of computations. And in addition, the length constant being so small for diffusion and the dendritic extent being quite long, you actually have a huge number of other compartments that you can use to carry out computations distinct from each other. Furthermore, in each of these compartments, there's a huge number of pathways. I'm being conservative. I'm saying there's roughly 100. You multiply it by the number of cells, and you end up with a lovely grand total of something like 10 to the power 17 operations per second. That's not even counting the slower terms for genetic computation. It's not even counting synaptic computations. So this is not bad at all. And in fact, according to this back of the envelope calculations, you're doing 100 times more computing with the chemistry in your brain than you're doing with the electricity in your brain. And both of those completely dwarf the number of operations, however complicated, are being done at the network level. Now, of course, this is a fictional subdivision of labor. And of course, all of these computations happen seamlessly across the scales that I showed you here. It happens, information flows seamlessly up and down these levels. So it's not really correct to say that it's happening chemically or electrically, because they're always talking to each other. Nevertheless, it gives you an idea of how important it is to be able to do the chemical computations that I'll be telling you about. Okay, so let me now home in on this question. How high level are these network operations? So, well, let's take it from the other point of view. What is the cell computing? So there's a whole bunch of things it does just on the broad, somewhat derogatory heading of housekeeping, but they're actually very interesting and quite complicated and quite ingenious, some of them. For example, the cell manages to stay alive, which is no mean feat. It handles all the incredibly complicated processing involved in development, going from a single cell to a complicated organism with all the network connectivity in the brain. It handles repair, and it does some very, very clever stuff to do load balancing within and outside every single neuron, which makes it possible, for example, for you to do fMRIs, because that is an outcome of the load balancing that takes place all the time. Okay, forget about those. Let's look at neural computation, and here's just a short list. I've certainly left out a whole bunch of possibilities, but here are some of them. The cell does various kinds of summations, spatiotemporal, it does EI balance, it handles neuromodulation. It does all sorts of very fascinating things with plasticity, association, maintenance, synaptic turnover, which is, I regard, as a very powerful form of plasticity because it allows you to form and remove connections. And with those, you really are in a ability to store a lot more information. It handles timing, and we'll be spending quite a bit of time looking at this, rhythms, pattern selectivity, and sequence computations. And then there's a whole bunch of speculative things which I probably won't be able to get into much detail about. Okay, so some of these are called high-level operations. And what I'm going to do is try to give you a glimpse, just a very brief glimpse of what these kinds of operations look like, and perhaps contrast how Neuro and how the cell goes about them. Okay, so let me start with this study, many studies, all of which say that spaced stimuli give better learning. So in this experiment, what Aziz et al. Did was they trained an animal in these bursts indicated by the gray bar, and they did it either in one intense massed section, longer spacing, more spacing, and more spacing. And not surprisingly, well, maybe surprisingly, the more the spacing, the better the animal's recall was. This axis should read recall, and this is, of course, first day before training and after training. So the more spacing there was, the better the learning. And this was done in rodents, but it's been seen in flies, it's been seen in worms, it's been seen in humans. The example I always give to students is that when do they learn better? If they have spent the night before the exam cramming, or if they have methodically spaced out their learning over the past few weeks and months. So they all say sheepishly, yes, but we only tried the cramming approach. So we don't know. Anyway, be that as it may, it's pretty clear that this is an interesting property of how brains learn things, that they learn better if there's spacing. Okay, so now there's various models for this, and I'll just give you a very brief glimpse of one of the kinds of models that has been done by Upinder et al. And this was quite a thorough model, quite an extensive model. It modeled the hippocampus, which is sketched over here. And their idea is that the network, the hippocampus here, has built in it cells which have certain preferences for time intervals. And in this complicated network of networks, so to speak, there's the hippocampus, nucleus accumbens, and so on, ventral tegmental area. So in this network of networks, you have information cycling around. And the basic idea, to cut a long story short, oh, we're having massive lightning. I hope the network isn't going to go down. Excuse me. Yeah. So, so the basic idea of this is that the any event that, excuse me, thunder happening. Okay, so the basic idea in this network model of interval learning is that when stimuli come in that map to a certain interval, then the appropriate cells contribute to synaptic change, And so the correct time cells increase their weight. When the interval is T1, a different set of cells would be reinforced for T2, and so on. So in this manner, you are able to learn and tune your responses to different intervals. Okay, so this is a way in which a network can achieve tuning to different kinds of intervals. So now, as I promised, I'm going to take you to the cell and see how it can do it. And so, oddly enough, inside the cell, we also have a network, but this is a network of signaling pathways. These are all molecules. I'm not going to ask you to decode the alphabet soup, but you probably know the NMDA receptor, which lets in calcium, you know the glutamate receptor, and so on. So the whole bunch of receptors, various effectors downstream. And anyway, you have a mess of chemistry happening. And the experiment that we did here, this was done by my student, Sriram AJ, many years ago. What he did was a very nice combined electrophysiology and biochemistry experiment. So what he did was he gave stimuli at different intervals. So here's the timing part. And here's an example stimulus where the interval is 600 seconds. So there's the stimulus. You get a small response, another stimulus, another response, and the third stimulus, you get a nice big response. So here you have spaced input. The stimuli are spaced apart by 600 seconds. And we tried this kind of stimulus for 20 seconds, 60 seconds, 600, and 1800 seconds. So there was a wide range of stimulus intervals that we tried. And what AJ found was, so here's the electrophysiology, let's focus here. He found that the plasticity that he measured in the electrophysiology was greatest at the 600-second interval. In other words, spaced was good. Massed was not good. When you cram all the stimuli close to each other, there was plasticity, but it wasn't so good. And interestingly, when the intervals were very, very long, very far apart, then again, the response was small, and you get small plasticity. Okay, so here's a tuning curve for interval, intertitanic interval, with a peak at around 600 seconds. So he also, over here, measured the biochemistry. He actually measured several molecules, but here we're going to look at the one that turned out significant. And this was MAP kinase, phospho-ERK2. And this one peaked at 600 seconds, which indeed coincides with the peak of electrical activity. And the computer model of this whole pathway predicted that, in fact, it should peak at around that time. So this is all very gratifying, that we had a model, we had electrophysiology, and we had biochemistry. The point here, though, is that all of this is happening within the cell. We're delivering a pattern of input, and it's being processed by a bunch of molecules, and it is giving us the exact same phenomenon, that is our preference for spaced stimuli, exact same phenomenon that this whole complicated neural network model was proposed to do. So here is an example then of a single cell doing a computation, a network computation, and accomplishing pretty much the same thing in, of course, a much smaller size. Okay, so I hope everything is still ticking along well. All right, so next, let me switch to sequence computations. And this is a topic I've been very interested in, because sequence computations are, I think, very fundamental to how the brain handles input. I'll give an illustration in a moment of a typical kind of sequence stimulus, and then we'll think about how this can be processed. But there are network approaches to analyze it. You can train neural networks. You can use SynFire chains. There's many subcellular approaches, which I will go through to give you a compare and contrast with some of the network approaches. But first, let's have an example of this. So I hope the movie comes across. If not, I will explain what's happening. So over here, this bluish blob is 2-photon imaging from a zebrafish. Now we've just zoomed out a little bit. We'll cycle around one more time. So here we have a zebrafish larva, I should say, and it's being imaged. And the bright streak of activity you see here are cells being highly active in the visual cortex. And what you may or may not be able to see is a little smaller streak zipping around over here. And what the smaller streak is, let's wait for the thing to cycle around to the slow motion version. The smaller streak is a paramecium. So there's a paramecium, it's just gone off screen. There it is again, it's swimming around. And the thing I want you to think about is, here is brain activity, yeah? And it is, I don't know if you can see the video, but the brain activity is sort of sloshing around in the cortex in a sequential manner. In other words, the visual, the sensory input that the brain perceives is being mapped onto the cortex, the visual cortex in this case, as a sequence of activation. This is not surprising, right? Because you have the topographic organization, you have the visual projections, which maintain the topography of the visual scene. But the important thing here is that here's a sensory input, and it takes the form of sequences of activity. And if you want, if, for example, if you're a fish and you want to go and eat that paramecium, then it's very much in your interests to be able to predict, to follow and predict where the sequence is going to take you next. And I will make the assertion that this is, in fact, a very, very fundamental, a very common computation that is done not just in the sensory physiology, but in many kinds of computation in the brain, whether it is language processing, whether it is navigating a street, whether it is simply thinking through a train of thought, which implicitly already has the implication of a sequence. So sequences are really, really important. So how can one process these? So here's a lovely example from Dean Buonomano's group. And in it, they had a recurrent neural network. And so here are the 100 of those units sampled, 100 of the neurons in the recurrent network. Here's time. And you can see it's kind of a mishmash. What's happening is that the neural network is being asked to learn a certain sequence of activity and try and predict what's going to happen next. And the sequence of activity that it is being given is a freehand rendition of two words, chaos and neuron. So what is freehand? Freehand is simply a sequence of x, y, and time coordinates for each point in the letter, in the word. Yeah. And so what the neural network has to do is to learn this sequence and try to predict what the next point is going to be. And I've not shown all the other details of this very nice study, but what it does is it starts out, completely all over the place. The sample points are the little dots over here, and the neural network is trying to follow them and predict what the next point is going to do. It does a miserable job. But after a little bit of training, it manages to follow the actual hand movement almost perfectly, as you can see by how tightly. So this is the final state of the network that has been trained. You can see how finely it follows the trajectory of the hand movement. So in other words, the network has learned a sequence, and quite an interesting and complicated sequence of XY positions, as well as the time associated with each position. So this is a form of network sequence computation, and quite a clever one. Okay, now here we come back to our hero, Wilfred Roll. And what Wilfred Roll did, getting on close to 50 years ago maybe, was to come up with a way in which a single neuron could do sequence recognition. And it goes like this. Supposing you have stimuli A, B, C, and D. If you give a stimulus at A, it takes a little time for that electrical impulse to reach the cell body. If you give it at B, it takes a little bit less time, and of course D is pretty quick because there's a very small distance to propagate. So what Wilfred Roll did was he computed how signals would propagate down the dendrite, and this is how they do that. So let's suppose we start with this one. So it's got a big response here to the stimulus. As the signal propagates down, this particular input starts to get shallower and broader, but it also starts to get a little bit delayed. Its time it takes a little while for the peak to reach, to build up over here. And so there's a delay, there's a propagation delay as it goes along. Now, here's the clever bit. If you deliver the next input, so this was input A, if you give input B at exactly the same time as the first signal is reaching its peak here, then you get a peak over here, adding on to the peak over there. And similarly, if this one is slightly delayed as well, its peak comes just when the other two have peaked over there, and so on. If you do this in the right order with the right interval, you will get a large peak at the soma. And this is the calculation that he did, which is that if you have it in the wrong order, then you get a shallow response. But if you have it in the correct order, then you get a nice big response at the cell body. So here, even a simple passive neuron is able to do a sequence computation. It's able to tell whether inputs are going in one order versus another. And it doesn't take very much imagination to think that maybe input A is one portion of the visual field, B is the next, C is the next, and so on. So in other words, you could have visual projections coming along in this, to this neuron, and then if an object was moving in this direction, then the cell would preferentially respond. If the object were moving in the other direction, the cell would not respond. So in principle, this single neuron is able to do a remarkable job of sequence computation, figuring out in which direction an object is moving, if it's connected up to the appropriate inputs. Now I'm trying to get this thing to, there we go. Okay, so that was simply using electrical passive responses, we are able to do sequence recognition. And let me just do my connectivity check. Okay, good. So let's move on. So Mike Hauser and Tiago Branco was actually the person who did the key work in his lab. They decided to actually look at this, to look at Rawls' approach, and they did it in experiments and in modeling to see if this would work at the single cell level. And they did a very elegant experiment. It came out about 10 years ago. And what they did was they recorded from a neuron, and they optically stimulated along a given dendrite. So here, zoomed in a little bit in panel B, you have the stimulus points 1, 2, 3, 4, 5, 6, 7, 8. And you can stimulate them in this order using glutamate uncaging, or you can stimulate it in the opposite order. In other words, pretty much doing what Wilfred Rawls had implemented back in the 1970 range. So fantastic. So now you can actually test out Wilfred Rawls' prediction. And Wilfred Rawls' prediction, if you recall, was that if you have a stimulus where the first input is there and the successive stimuli go towards the cell body, in other words, the in arrow, then you should get a larger response than if they go in the opposite direction. And sure enough, this is what Tiago Branco and his colleagues found, Mike Hauser and his colleagues found, which is that the in direction, the red line, had a larger response than the out direction, and this was a function of the input velocity. So it wasn't just selective for the direction, it was also selective for the velocity of the sequence. So quite a good computation, I would say, for a small piece of a single cell. They analyzed it further. They showed that the speed range was most effective in this a few microns per millisecond, and that's where you have the greatest selectivity at about 3 or so microns per millisecond. So this is actually pretty fast. Yeah, if you convert this into how long it takes to go over the entire length, which is roughly 70, 80 microns, then that's over in of the order of a few tens of milliseconds. So this is pretty fast sequences. It would be an object moving really fast, say, across a field of view. Okay, so this is nice. They went further. They looked at the mechanisms. They established that calcium was coming in large amounts when it was the inward sequence, in other words, the preferred direction, and much smaller amounts of calcium, which is the bright red in this case. So the outward version has only yellows, which means that it's not a strong response. And at different speeds, they get different levels of calcium in the different cases. So this is the preferred speed. And here's another speed where you also have quite strong discrimination. And in both cases, you can see there's much more calcium in the in direction than there is in the out direction. So they went further. They said they put in an NMDA blocker, NMDA receptor blocker, APV, and showed that then the selectivity went away. And to cut a long story short, they replicated Rawls' predictions, but they added the wrinkle that a significant part of the selectivity came because the NMDA receptor is voltage dependent as well as ligand dependent, and this gave a boost to the selectivity that Wilfred Rawls had predicted. Okay, good. So here we have another way of doing sequence selection. I'm going to pass very briefly over this other very beautiful study on amacrine cells, on starburst amacrine cells, where again one gets sequence selectivity, and this is very direct visual input, right? Amacrine cells are in your retina. And what they found, Ding et al. Found, was that there's an extraordinarily precise pattern of inputs, inhibitory versus excitatory, over the dendritic arbor of the neuron. And because of this selectivity, because of this patterning of inputs, the organization of inputs, these cells are selective for inputs going in a certain direction. I won't go into the details of this in the interest of time, but this I thought was an actually elegant demonstration of sequence selectivity. Let me now focus on my own favorite version of sequence selectivity, which is biochemical. So the idea is actually very simple, similar to what Wilfred Rawls had, except now think about it in the chemical domain. Supposing you have inputs from different sensory inputs, let's say different visual locations, 1, 2, 3, 4, 5. Supposing these inputs come to different locations in order on the dendritic trees, they come to different spines. Now, instead of electrical activity propagating, we have a chemical signal which builds up underneath the first spine, and then it spreads, just like the electrical activity would spread. And if you come along and give it a boost at the right time, at the right place, you can build up your response. And conversely, if your order is scrambled, if you go back and forth, then your inputs come in, same inputs, but they don't build up. They don't build up because they're not reinforcing each other as they go along. And to analyze this, we started out with a spherical cow model. A spherical cow model is one which has the minimal possible number of variables to keep it really, really simple. Of course, this means that it has about the same relationship to neurons as a spherical cow does to the real kind. But we start with spherical cow model to make those who like abstract models happy. So I tried a number of spherical cow models. These are different spherical cows. I'll just focus on this one. So here's basically what we did. We gave a calcium stimulus. I hope the movie is showing. So you give a calcium stimulus which trundles along the dendrite, and then the model, a whole bunch of signaling pathways are localized at specific places, and these build up if the stimulus is in the right order and do not build up if the stimulus is scrambled. So let me illustrate this. So what you're going to see now, and I hope this comes across. Oh, wait, here's the static version. So in the correct order, you get a nice strong buildup of responses. In scrambled orders, you get a very small response. So this is the sequence this is the response in sequence, and this is these two are examples of the response out of sequence. So let's see if we are able to see this movie. Please, somebody put it on the chat so that I can tell if the movie has worked. So focus on the blue curve first. That's the sequential input, right? So the input is coming in order, and you get a nice big response building up. The scrambled one, which was in red, hardly builds up at all. I'll run it by you again. There's the blue. You can see it's going in order, click up and up. So you have a huge response here, and it slowly collapses after the stimulus is gone. Exactly the same strength of input. Now we focus on the red. Is there, 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 and there. And we don't get any buildup at all.
The movie's working much more beautifully than we tested it.
Okay, very good. So I'm glad that it's working. So you can see then that identical amplitudes of inputs, it's just that one is in sequence and one is not, and you get a build-up if it is in sequence. And this is just happening in the chemical domain. Okay, now, of course, the neuron is not a spherical cow. This is, to my mind, this is a really, really terrifying picture. It's a beautiful graphic from Sven Truckenbrot. And what it is a single presynaptic bouton. What's terrifying about it is it gives you a sense of just how densely packed with proteins, with computational elements, if you like, is the presynaptic bouton. It's just jam-packed with proteins. There's the synaptic release zone. So this is the non-spherical cow version. This is what the real chemistry in the cell looks like. It's just incredibly dense, compressed version of computational machinery. You can sort of see a few microtubules sticking around there, a few vesicles in there. It's just wonderful how much computation you could do with this thing. So what can you do with this? A slightly less detailed thing than this thing is what the model that we built, which was a 3,000 compartment model with 5,000 spines. I've illustrated the little spines, which is what you can see here. Each spine in this model has the full set of reactions. I won't go into the details of that, but several tens of reactions and molecules in there. There's background synaptic activity of this whole thing. And the challenge for this cell is to pick out those Z regions on the dendrite which receive sequential input. It's random activity all over the place. There's sequential activity only in just a few places. So I've been assured that this movie does not actually come across very well. But if you could see what I'm seeing, what you would see on this neuron is little spots of light, which represent synaptic input coming randomly. And this is like little flickering lights flashing on and off over the entire cell body. And I'm sorry that it is probably past the bandwidth capacity of my network to illustrate in all its glory. So, but this is what neurons have to contend with. Little inputs coming all over the place, thousands and thousands of them over the cell. Okay, so the assertion I'm going to make is that tiny little sections of this dendrite, 30 micron sections, can carry out sequence discrimination. I'm going to hazard another movie. This one is a neuron. I've zoomed in onto the portion of dendrite which is getting the sequential input. And as you may be able to see, it's all gone red. I'll just run it by you again. So red means there's a strong response. And if you're able to see it, the inputs are coming in sequential order and it builds up very strongly. If we switch over to the scrambled side, the inputs here are jumping around and the net result is blue. In other words, a very weak cellular response for the out of order sequence. So even if we put in the details, the sequential selectivity remains. Okay, so I hope that at least some of that movie came across. Good. Thank you for those who've helpfully told me whether or not the movies are working. Okay, so now just a quick recap. We've looked at the network level of how you can use a network, and this was Dean Buonamano's analysis of how a network can be used to recognize a sequence. This thing worked on the order of a 1 second time scale. We saw a subcellular version of this, Wilfried Roll and Mike Hauser's analysis. This worked at the order of 100 milliseconds. And then the chemical version, which covers everything from calcium-induced calcium release, which is fast, to other kinds of signaling, which is slow. And so this is of the order of 0.2 to 10 seconds. So this is very much in the behavioral time range. This is quite fast, but there's certainly some kinds of things for which this would be applicable. And this is also in the behavioral time range. But the important thing is that this huge network is recognizing sequences, but so are these tiny pieces of single neurons. Okay, so we've gone through this list of things. We've seen some network approaches to sequence recognition, and we've seen some subcellular approaches where the Ramon y Cajal's butterflies are doing pretty much the same thing, except very efficiently. So now I'm going to get completely speculative, and I'm going to take you to neural networks again. So how do you get sequences into a neural network? So to get a sequence into a network, you have to have time information. You have to have history information. The network has to know what happened at previous time points. And in a classical neural network, you don't have that, because you just have the input and what propagates through as the output. There's no time domain. A clever trick to introduce time, introduce history into neural network is to make it a recurrent one. In other words, some of the output signals go back into the input and thereby provide context about what the neural network was thinking about in the previous time step. So this is what deep networks do. They take a recurrent neural network, and instead of, because they have massively parallel computers, what they do is they unroll this. So basically, you take the network at time 1, and you take the network at time 2. So network at time 2 is the same network, really. It's the same network, but it's what they do is they simply make a copy of the network and give it the input pertaining to the other time. And this is what's called unrolling the network. So they have a lot of copies of the same network for each time step that they're analyzing. And this is how the deep networks, this is why they get so deep, because they're including a lot of history. They do a number of other clever things. For example, each of these networks, which has got a whole bunch of neurons and synapses in it, is replaced by one node of something called a long short-term memory. I don't know why they came up with that name. LSTMs are one of the standard building blocks for this. But they have these, and then they put these in nodes, and they make these nodes into deep networks, and so on and so forth, right? And the important thing about LSTMs is that they have an internal state. In other words, they remember past events. So I think you might be able to see where we're going with this. These kinds of things, because they have internal state, they are able to process sequences, and they can do quite remarkable things. So I don't know if you can read this, but this is the output of a neural network trained on the complete works of Shakespeare. And although the text is complete garbage, if you were to read it out, it actually has the same rhythm and almost the same sentence structure as you might imagine Shakespeare writing. Alas, I think he shall become approached, and the day when little strain will be attained into never being fed, and so on and so forth. So this network has somehow imbibed some of the sequential properties of William Shakespeare's writing, which I think is quite remarkable. So, okay, so can we use our capability for single neurons to do sequence recognition and apply them to this? So this is where I'm really going out on a limb and being speculative, but here we go, yeah? So subcellular sequence computation is a building block. It is something we've seen happening at many levels with many mechanisms in real neurons and in model neurons. And there's a huge number of possible sequences a single neuron could recognize because it's got thousands and thousands of synaptic inputs. So what can it do? Every neuron, every dendritic tree can do summation, it can do nonlinearity, and it can do so for tens of thousands of inputs. It has all the interesting properties you want of gating, of control of which input is relevant at a given time. EI balance, for example, can allow only one of these many possible inputs to be selected. And importantly, each one of these pieces of dendrite is able to recognize these these pretty colored sequences. So each portion of dendrite we're proposing is capable of recognizing sequences. And therefore, in parallel, the neuron is processing, in principle, and recognizing thousands and thousands of sequences at the same time. And on top of this, the neuron is able to learn and modify its responses to each of these sequences. So what I'm going to speculate is that, at least in terms of some of the capabilities, the neuron is doing at least as much as one of these LSTM, one of these deep neural network nodes. And so with this, you are able to do in one cell the same kinds of time and sequence recognition, the same kind of learning and state information throughout the cell. Unlike this kind of network, though, a neuron typically has only one output, so the analogy isn't perfect, but I think it's very suggestive. So to summarize then, the single neuron is doing an incredible amount of computation. Some of it is clearly not the kind of thing that you really want to afflict upon a network. Some of it seems rather too simplistic for a network, but I would make the case that a lot of very, very interesting things in this section and maybe things which I haven't even thought about or touched upon are being computed by single neurons, including what I've shown you, intervals and pattern selectivity and sequence selectivity. And who knows, maybe even things like deep neural networks do long short-term memories and so on. We have a current project which suggests that single neurons through dendritic patterns can even do some novelty detection. So these are, I think, really interesting possibilities for single neurons to take. Okay, so then to wrap up, networks are a dominant metaphor for brain computing, but I'm making the case that some high-level computations can be done within a cell, including pattern recognition, sequence computation, and probably many more. So single neurons have these capabilities just like perhaps deep network nodes, but single neurons are tiny and incredibly efficient. And this, I think, is what makes it possible if we take the idea that at least some kinds of computation that the brain is doing is analogous to what these gigantic server farms are doing. This is what makes it possible to do it in the small volume of your head and with an incredibly low energy footprint. And so to come back to Ramon y Cajal, this is what I think are the computations that the butterflies of the soul are doing. Okay, so thank you all very much. This is a glimpse of my lab, who have provided me much of the inspiration for doing all of this. And hopefully in happier times, we will all be able to travel again. And if so, please do come and visit us. Thank you very much.
Clapping for the 100+ people in the room. Thank you very much. We have some time for questions, and we do have a few questions. I'm going to invite some of you on screen and see if you're going to pick up the invitation.
Okay, yeah, so there's a question about Numenta sequence prediction. Yeah, I should have brought that up. So Numenta has been thinking about this, and I actually had a chat with one of the people involved in that work. I think that, yes, this goes a little bit further because their analysis typically went for very short sequences, a couple of inputs. But I think that this could go for much, in fact, it's more interesting when you have longer sequences because then the selectivity becomes more computationally interesting. It becomes much tighter.
So the top voted question was about complexity of each of the individual operations on the network level versus electrical or chemical level. Dan is wondering or imagining that a network level operation processes much more information and can be much more complex than an electrical or a chemical one. Would you copy that, or?
So yes, of course, the network is, through parallelism, it is representing in principle much more complicated concepts, if you like. And so I'm completely on board with that. But the case I'm trying to make, though, is that some of the computations which have been ascribed to network level operations should really be moved to the subcellular level. And the network in this way of thinking is directing the inputs through the connectivity. It's directing the inputs onto the neuron where the inputs will do most good.
Yeah. Venkat, I'm going to invite you to the screen. I'm going to invite, I'm going to close your screen, if that's okay with you. Let's see if we have people joining us. Yeah.
Hi, Venkat.
Hi, Upi. Great talk and great to see you. My question is the following. So Tony Bell talks about a notion of submergence. It's the idea that information percolates down the scales to the molecular/chemical level, where computations happen, and this could sort of bubble back up via emergence. And this sort of interplay of submergence and emergence is central to computation in the brain. And of course, as you point out, explorations of chemical computation are already quite underrepresented. So do you see promise in this sort of holistic dance of scales, as it were?
Well, absolutely. I mean, I think it's something I pointed out right at the beginning, which is that it's a mistake to restrict one's analysis to the upper or the lower level of brain function. The flow of information is continuous and bidirectional, and it's absolutely integral to how the brain works. It's not a good idea to say that it's only subcellular or only electrical or only chemical.
Thank you.
All right, another question. If we were to make a parsimonious computational model of the brain, which comes close to mimicking its function, would it include lesser components? Would it include more components than the real brain or fewer? To clarify, would we need the same number of variables as the number of cells to make that model?
Oh, that's a really tough one. It depends what you mean by mimicking the computations of the brain. And I mean, this is a sort of the reference, the standard question in modeling. How much detail do you need to put in? And the answer is always, what is the question you want to ask? So if your question is something on the lines of what drugs do I give to help repair the brain after injury, then you have no choice but to start to look at the molecular level. If your question is how do, say, linguistic concepts get represented, then I would make a speculation that you would want to have something with a strong basis of sequence computation, but you could do this at quite an abstract level. But this is speculation. We have seen existence proofs in that, for example, various artificial intelligence programs are able to do a reasonable job of being to interpret what people are saying. And they're doing this at a fairly high level of sequence analysis and putting together, trying to infer meaning from that. So that would be the appropriate level, I think, of representation for that question.
And to get the whole shebang, to model the entire brain and its complete function.
I mean, if you want to model the whole brain to do everything the brain does, you need a brain. I mean, I don't think I think it's an ill-posed question, right? You want it to replicate everything the brain does at all levels, then you need the original. You need everything.
All right. Dean, next question.
Hi. I was wondering about the speed at which a signal propagates down the dendrite. It seems intuitively that it sets a scale at which we can be sensitive to the speeds in stimuli. So I was wondering if there was maybe any kind of hypothesis about a evolutionary relationship between the speed at which a signal can propagate down the dendrite and statistics of natural stimuli.
Yeah, that's interesting. I mean, the zero order answer is that yes, I think some of the chemical mechanisms, in particular calcium-induced calcium release, is on the somewhat faster end of the scale of behaviorally relevant events. And some of the purely chemical ones are on the slower end of the behavioral scale. And the electrical computations that Mike Hauser and colleagues were showing, that's on the really fast behavioral scale. I think you need all of these because humans are actually quite slow when it comes to processing real-world inputs. You just have to see what a mouse or even an insect, which they work at incredibly short time scales. So the kinds of computations that we think are behaviorally relevant are actually very slow for them. So I think you need all of these time scales. You need to be able to do things really, really fast. And sequence detection, I'm sure, is happening in all of these levels, in all of these organisms. And I think nature is very parsimonious. It's probably using all of these mechanisms.
Thank you.
All right, a question from Tom Burns. Asks states that you seem to focus on calcium modeling in dendrites. Functionally, how is that a more appropriate thing to model than electrical charge in such dendrites? What other chemistry molecular biology processes do you think are worth modeling, and which holds the sort of computational power you intimated via a large number of operations per second compared to electrical network scales?
Okay, so the way I think about it is, what are the kinds of time scales you would get out of different kinds of mechanisms underlying it? Because we've seen that there are several ways of doing it. So the electrical time scales are sub-second. They're of the order of 100 milliseconds. That involves one set of mechanisms, if you wish, the NMDA receptor being important among that. I've looked a lot at the calcium signaling because that is operating in the time domain, which I happen to think is interesting, the half a second to a second or two time range, calcium-induced calcium release. But the model, in fact, the movie I showed you of the Dendrite was operating on a still slower time scale of kinases and phosphatases and second messengers. So we model all of these things. We model the ion channels, we model the calcium, we model the signaling pathways. And that's why I've got the sense that there's this range of domains of timescales which these different mechanisms can handle. And it's actually, I think, interesting and important. And presumably the nervous system is using all of them.
All right, 2 more questions. When we move from single neurons to a network of neurons, would there be a delay in terms of the operations? If so, what do we know about this?
When we move from a single neuron to a network, is there a delay? Well, I mean, single neurons, their computational operations are the elementary units of what the network is doing. So when you have, for example, an action potential coming out of a neuron, that speed of propagation is just what the network will be able to convey to the next layer of the network. So I don't see that there's any particular delay in that sense. Where you may have a delay, you probably have a delay, is the information coming in to the single neuron being acted on in some way, and that finally manifesting as an action potential or change in firing rate. So that time is of the order of the time scales I have mentioned earlier, which is the different time windows for electrical, calcium, or biochemical signaling.
Yeah. All right. Thank you, Upinder. I think that's it. I think we'll leave it at that. And I would like to thank you again for this lovely and thought-provoking talk.
Thank you very much.
And thanks to the audience for coming by.
Thank you to the audience.
Lovely Friday.
See you all.
And I'll see you on the other side. So goodbye for now.
Okay. Okay.